Hello, everyone, and thank you for coming to our July meeting. So I have a couple of announcements before we introduce our first speaker, and that is this month's Myco Talk is on July 29th. And so those are highly recommended. And the speakers are Duncan Wilson and Jules Trainee, and I'll put the registration info into the chat. The other announcement is that Dr. John Norman from the Global Mycology Institute and Global Vaccine Center is looking for postdocs interested in using precondidial mycelia and other fungal products in vaccine development, as well as an emeritus mycologist interested in leading the mycology aspect of this institute. And I will put John's contact info as well as the website for this institute into the chat if anyone is interested in investigating those opportunities. And with that, I will pass it off to Cheng Ben, who will introduce Jesus Romo, our first speaker. Great, thank you very much. And there are lots of questions in the chat, so please keep putting more questions in and we'll take them all at the end. And our second speaker today is Sébastien Ortiz, who is a PhD candidate who's almost finished his work in Christina Hull's lab at the University of Wisconsin-Madison. So Sébastien received his BS in chemistry at the University of Virginia. He's the 2017 class of the Howard Hughes Medical Institute's Gilliam Fellows Program, which is a fantastic program for underrepresented postdocs that I recommend to anyone. And in September, he will be joining the Bertuzzi lab in the Manchester Fungal Infection Group to start his postdoc. And so let's hear about your wonderful PhD work and best of luck in your postdoc. So please keep entering questions in the chat.
Thank you so much for that introduction. Yeah, so today, can you hear me okay? Can you see everything okay? Wonderful. Thank you for this opportunity. Today, I'm excited to tell you about how we're exploiting Cryptococcus spore germination as an untapped source of fungal-specific targets. So this audience in particular, you all know that invasive fungal pathogens are a problem. They cause around 1.5 million deaths a year. And part of the reasons that this burden is so high is that we have limited antifungal therapeutics available. Really, they come in two flavors. Our first option is amphotericin B, which is extremely effective at killing fungi, but also extremely toxic. Alternatively, we can use other antifungals that are not very toxic to humans, but they're not as effective for a variety of reasons. And so currently, the gold standard for many invasive fungal pathogens, including Cryptococcus, is amphotericin B, which is rather pathetic considering some clinicians have nicknamed it amphoterrible, and it was developed in the 50s. So we can be doing better. In addition to this, antifungal resistance emergence has occurred globally. This is a review by Matt Fisher and colleagues that shows all the different mechanisms by which fungi have developed resistance against the antifungals we use both in humans and animals, but also the fungicides we use against plant pathogens. And this is rather alarming because as resistance mechanisms emerge, our arsenal to combat human fungal pathogens becomes weaker and weaker. So one of the big hurdles in developing antifungals is that fungi and humans are both eukaryotic. And so this causes some problems. And I think this XKCD comic really encapsulates that. But because we're both eukaryotic, we look alike molecularly. And so when we inhibit processes important for fungi to grow, we often inhibit processes for our own cells to function, leading to high toxicity. So what we need to do is we need to target fungal specific pathways. Unfortunately, the current targets we have are not quite sufficient. So I'm going to walk through our current targets. Here's a yeast cell. And so one of our most common targets is ergosterol, either itself or its synthesis through polyenes and triazoles. So that's the cell membrane. Alternatively, we have echinocandins, which inhibit the synthesis of beta-1,3-glucan, so the cell wall. And then we have flucytosine, which inhibits nucleic acid synthesis, but it can't be used as a monotherapy, so that's not great. And really, this encapsulates our targets currently that we use. That's not great. We need new fungal-specific targets. But how do we identify these? Well, one idea is that we can exploit fungal-specific differentiation processes because given their fungal-specific nature, they likely have molecular pathways that are specifically evolved for fungi. And so we've decided to target spore germination. Spores are a dormant stress-resistant cell type used by fungi to spread to new environments. And spores are important infectious propagules both in plant pathogens and human pathogens. And when spores encounter the right environment, like let's say the mammalian lung, they can escape dormancy and undergo this differentiation process called germination, where they become a vegetatively growing cell. So here you see a spore, an SEM of a Cryptococcus spore germinating into a vegetatively growing yeast. And so we study in the whole lab spore germination of Cryptococcus for a variety of reasons. But one of the reasons is that we hypothesize that spore germination holds fungal-specific molecular pathways. And these pathways could be ideal in the development of novel antifungals. Additionally, spores are important infectious propagules for fungal disease. A variety of human fungal pathogens make spores. I've told you Cryptococcus makes spores, the molds Aspergillus fumigatus and the Rhizopus oryzae make spores. The fungal dimorphs, histoplasma, coccidioides, and blastomyces all make spores. In fact, the majority of invasive fungal pathogens are spore formers. And so these are important infectious propagules for us to study. Additionally, spores can demonstrate unique disease kinetics. So we've seen this in Cryptococcus specifically. So here's a beautiful SEM by a postdoc in our lab, Eddie Dominguez. And it's an image of a macrophage uptaking a cluster of Cryptococcus spores. And we've shown that spores are readily phagocytosed by macrophages, much more so than yeast. And in fact, as spores germinate into yeast and become more yeast-like, their ability to be uptaken by macrophages goes down. And this preferential phagocytosis of spores has been directly linked with the ability of spores to disseminate out of the lung and into other organs. And so if we could understand germination, we may be able to modulate disease kinetics of spore-mediated disease. Additionally, it's important to note that spores have to germinate to cause disease. So spores can undergo germination, and once they germinate, they can start replicating. However, if spores cannot replicate by themselves, they have to first undergo germination before they can replicate. And so when they cause infections, they have to cause an overwhelming fungal burden, in this case in the brain, which is how cryptococcosis presents itself usually. Spores have to be able to germinate into yeast, because if they can't become yeast, then they can't replicate. And if they can't replicate, they can't cause an overwhelming fungal burden. And so germination is required for spore-mediated disease. All right, so we study Cryptococcus spore germination for a variety of reasons. One of them is to identify novel fungal-specific targets. Another is in the hope that we might be able to prevent spore-mediated disease. And finally, to characterize novel fungal molecular biology. So spore germination is poorly characterized in all fungal systems. And so there is unique biology there for us to probe and learn about eukaryotic differentiation. And so today I'm going to focus on the identification of fungal-specific targets using novel inhibitors. But not a lot is known about spore germination in any fungal system, and there aren't a lot of tools to study spore germination. And so we had to create our own tools. However, how do you study something you know almost nothing about? Well, what we do know is that spores are small and oval and yeast are large and round. And we can take advantage of this morphological transition to quantify the process of germination. So we developed a microscopy-based quantitative assay where we can load spores into a well, image them, and then measure the size and shape of each individual cell. And we can monitor the process of undergoing this morphological change for the same population over time, essentially doing single-cell microscopy on a population level. And we can quantify all of this. So I'm going to be showing a lot of two-dimensional histograms today, so I'm going to explain them here. On the x-axis is area, so a measure of size. On the y-axis is aspect ratio, a measure of circularity. So spores which are small and oval are in this lower left-hand quadrant, as seen here. And so the brighter the pixel, the more cells fall under that size and shape category. As spores germinate, they become more circular and larger and transition to this upper right-hand quadrant as they become yeast cells, as seen here. And so we can monitor this transition. So here is a population of roughly 10,000 spores at the 0-hour time point. And we can monitor this exact same population over time in nutrient-rich conditions. And what you can see is that the population of spores synchronously becomes more circular and larger as it becomes yeast. And so we can monitor the population dynamics of these cells. But not only that, this is all quantitative. So here is the exact same data, but in a stacked bar plot. So on the x-axis is hours of germination. On the y-axis is the percentage of the population, and spores are in orange, yeast are in blue. And as you can see, as germination occurs, the percent of the spore population goes down while the population of yeast goes up. Okay, now that we have this tool, what can we do? We can look at deletion and overexpression constructs of genes that we think might be of interest. We can look at nutrient requirements of germination. We can couple this assay with fluorescence, which can allow us to probe specific molecular pathways of interest. And we can also test inhibitors of germination. And we can use these inhibitors of germination to identify novel targets. So I'm going to focus on these inhibitors today. What I haven't told you about this assay much about is that we can look at population behaviors in a very detailed manner. So I'm going to use alexidine, an FDA-approved drug that we identified as an example. And I'm just going to show you 0 and 16 hours. So most inhibitors at low enough concentrations don't inhibit germination. That makes sense. And so what you can see is that a population of spores fully germinates into yeast. At high enough concentrations, most inhibitors fully inhibit germination, as seen here by a complete lack of transition out of the spore state. And so, but what's really interesting is the intermediate concentrations. What happens in between these? And so what we can see is that there's a unique population dynamic happening at this intermediate concentration. And in fact, different inhibitors demonstrate different population behaviors. Different mutants demonstrate different population behaviors, and different nutrients do as well. And so we were interested in characterizing these phenotypes. So we used a very well-characterized inhibitor, cycloheximide, which inhibits protein translation. And we know from other studies that fungal spores can't germinate without protein translation occurring. And so here you see a titration of cycloheximide from 80 micromolars down to 0.08 micromolars. And you can see we go from full inhibition of germination all the way down to no inhibition. But what we're really interested in is this intermediate space. So at these intermediate concentrations, what are the spores doing? How do they behave as a population? So I'm going to show you the two-dimensional histograms here. Here is our control, 0 micromolar. And as you can see, spores germinate synchronously over 16 hours. When you add cycloheximide, the population of spores remains synchronous, but they slow down. And as you increase the concentration, they slow down even more. Now, I know these plots are hard to look at if you don't look at them every day like I do. So I'm going to highlight these boxes. What you can see is that the population of spores at 8 hours at 0 micromolar is nearly identical to the population at 12 hours at 0.6 micromolar, which is nearly identical to the population at 16 hours at 1.25 micromolar. And as we increase the concentration, germination slows down more and more until it fully stops. And we can quantify all of this. So here is a rate plot. So on the x-axis is the time point of germination. On the y-axis is germination rate. And what you can see here is the rate of germination. So for no cycloheximide, you see this bell curve, which is canonical for germination. And as you increase the concentration of cycloheximide, the spores are germinating slower and slower. So we know that we can have this synchronous slowdown when we inhibit translation using cycloheximide. So in theory, if we were to inhibit translation with other inhibitors other than cycloheximide that target translation, we should again see the same behavior by the spores. And so our hypothesis was that other translation inhibitors would give us the exact same behavior. And so we tested this. So here's geneticin, which inhibits peptide elongation. And I'm just showing you 0, 8, and 16 hours for simplicity. And in the presence of geneticin, again, we see this slow down phenotype. We tested other translation inhibitors, anisomycin, again, slow down phenotype. Puromycin, even though it required a lot higher concentration, again, showed this slow down phenotype. And so this suggests that if you inhibit the same process, you get the same phenotype. And in fact, this is something we've seen for multiple different cases where inhibitors of the same process or mutants of the same process lead to the same population behavior. So now that we know that inhibitors that target the same process lead to the same behavior, how many of these phenotypes are there and can we discern/quantify them? So long story short, we've identified 6 distinct germination phenotypes so far. And the first way we can discern them is germination still synchronous? And this gives us 2 categories. First, if they are still synchronous when inhibited, they fall under this homogeneous germination category. Alternatively, if they're no longer synchronous, they fall under this heterogeneous germination category. And within each of these, we have 3 phenotypes. For homogeneous germination, we can discern them based on rates. So I've already told you about the slowdown phenotype with the translation inhibitors, but there's also slow start and slow end, which is exactly what they sound like. We didn't come up with complicated names. So a slow start, they're slow to start germination, slow end, they germinate and then they stall out. And we can quantify this based on rates. So here's another rate curve, just like the ones I showed before. And again, this canonical bell curve for the control. Slow down, just like with what I showed you with cycloheximide, leads to this prolonged bell curved. Slow start, on the other hand, you see very little germination at the beginning, and then the spores overcome inhibition and start germinating faster. On the other hand, with slow end, they germinate normally, and then they start stalling out at the end. And so we can discern each of these and quantify them. Then there's the heterogeneous germination phenotypes, and these are more complicated in that it's based on distinct population dynamics. So we have asynchrony, bimodal, and morphological variability, and I'm going to describe each of these. So here's our control, just DMSO. So what you see is that spores are germinating synchronously over the course of germination. And in the presence of inhibitors of each of these phenotypes, we see unique population dynamics. So in the case of asynchrony, what we see is that each spore is inhibited differently, resulting in this wide spread of different germination rates across the population. Alternatively, we can have a bimodal phenotype where one part of the population of spores doesn't germinate, and another population of spores germinates normally, leading to two populations. Finally, there's the morphological variability phenotype, where spores germinate into yeast, but these yeast have a wide variety of sizes and shapes. And again, we can quantify this based on the population dynamics. So these plots on the x-axis have shapes, so area times aspect ratio, and the y-axis is the percent of the cell population. And so I'm just going to point out bimodal because it's the easiest to conceptualize. At 16 hours, you see that there are two distinct peaks, one that resembles spores and one that resembles yeast. And so we can now quantify each of these phenotypes. Okay, now that we've defined these, what can we do? And can we use these phenotypes in target identification? Well, to do target identification, we first need novel inhibitors. So lucky for us, we had Mingwei Huang in the lab, who was a postdoc in the lab. And he developed a high throughput screen that is based on a protein luciferase construct, where spores don't emit a lot of light. And as they germinate, they emit more light. And so what you can do is in a very high throughput manner in a 384 well plate, measure light output and identify putative germination inhibitors. And so we screened a variety of libraries. We first screened an FDA-approved drug libraries in the hopes of potentially repurposing already approved drugs. And I'm not going to talk about that here, but it's been published if you're interested. And then we also screened 3 libraries of structurally diverse compounds that comprise 75,000 small molecules. And so we screened these molecules, identified putative germination inhibitors, and then put them through our quantitative germination assay. And we identified, confirmed, and characterized 191 novel inhibitors of spore germination. And we also coupled this assay with optical density readings, so of replicating yeast. So we also found that most of these also inhibited yeast replication. To our delight, 76 of these inhibitors fell into 8 distinct structural groups. So we went from 75,000 structurally diverse compounds to compounds that resemble each other. And this is a benefit to us because if compounds are similarly structured, then they may be inhibiting fungal germination and growth in a similar manner. And so we can move forward with them as a group, hypothesizing that they're targeting the same thing. Additionally, 63% of the inhibitors we identified had low cytotoxicity against mammalian fibroblasts. So this suggests that they're inhibiting Cryptococcus in a fungal-specific manner. And so I'm going to be focusing mostly on this subgroup. Group B is what we call it. It's a furan carboxamide, mostly so that you have a consistent story. So we hypothesized that compounds with similar structures would have a similar phenotype, right? Because they're likely targeting the same thing. And we know that in the cases we've looked at, if we're inhibiting the same process, we get the same population behavior. So here are 10 compounds, the 10 compounds we found in this group, organized from strongest to weakest. And I'm just going to show you one concentration, one time point that shows this, the phenotype for each of these compounds. And so what we found was that every single one of these inhibitors showed this asynchrony phenotype, which was great. It suggests that they're all targeting the same thing. And this trend generally held true across other structural groups. So the more structurally similar a compound was, the more likely it was to have the same phenotype. And since we're focusing on group B, most of these inhibitors had relatively low cytotoxicity at these concentrations, suggesting that they're likely inhibiting Cryptococcus in a fungal specific manner. So I've shown you that shared targets lead to shared phenotypes. We know that shared structures often have, compounds with shared structures often have shared targets. I've also shown you that shared structures lead to shared phenotypes. Now, can we use these shared phenotypes to help us infer targets? So again, here's group B, the furan carboxamides. I've told you that they all have this asynchrony phenotype, that they have a wide range of potencies, both against spores and yeast, and they have low cytotoxicity against human dermal fibroblasts. And so the one thing I haven't told you yet is that they have this shared substructure with known SDHI fungicides. And I'm showing you an example here of furcarbonyl, one of these SDHI fungicides. And so an SDHI fungicide is a succinate dehydrogenase inhibitor. And for those of you who don't remember this, because you don't study the electron transport chain, succinate dehydrogenase is complex 2 of the electron transport chain. And SDHI fungicides are used against plant pathogens by inhibiting this process. And SDHI fungicides are a wide range of different compounds, of different structures that inhibit this process. And a small subset of them have this same furan carboxamide substructure. All of these have been discontinued for a variety of reasons, but this led us to the hypothesis that our group B compounds were likely targeting succinate dehydrogenase. Well, if this is true, then these SDHI fungicides would also give us the same asynchrony phenotype. And so here I show you a B1, our weakest group B inhibitor, which shows this asynchrony phenotype at 80 micromolar. Furcarbonyl, one of these SDHI fungicides, is very similar to B1 in that it's only missing this ethyl group right here. And so, just like we suspected, furcarbonyl showed this exact same asynchrony phenotype at 80 micromolar, suggesting that they may be inhibiting Cryptococcus in the same way. Okay, so if our group B inhibitors are inhibiting the electron transport chain, then we would expect other electron transport chain inhibitors to also give us this asynchrony phenotype. And so that's what we did. We tested a few electron transport chain inhibitors. So here's furcarbonyl, an inhibitor of complex 2. And we tested rotenone, TTFA, and antimycin A, which inhibit complex 1, 2, and 3 respectively. And what we got was, again, this asynchrony phenotype for each of these, suggesting that we may be inhibiting this electron transport chain. We then decided to look at oxygen consumption. So we know that we can measure oxygen consumption using these seahorse assays. And so if we're inhibiting the electron transport chain, then we'll lower oxygen consumption. So on the x-axis is time, on the y-axis is oxygen consumption rate, and each of these is an injection of a different compound. So yeast, without glucose present, don't consume a lot of oxygen. You add glucose, the oxygen consumption goes up. And then if you add furcarbonyl, this SDHI fungicide, oxygen consumption goes down. And then Cryptococcus yeast is able to overcome this inhibition. And then if you inhibit complex 1 and 3, you can bring it all the way down. Well, this isn't too surprising since furcarbonyl is not a particularly good inhibitor of Cryptococcus. But what about one of our promising inhibitors, B2? And so we tested B2, and again, low oxygen consumption rate, you add glucose, oxygen consumption goes up. And then if you add B2, then oxygen consumption goes back down, suggesting that we are inhibiting the electron transport chain. So altogether, all of this data supports the idea that our novel furan carboxamide inhibitors are likely targeting complex 2 of the electron transport chain. And because of their low cytotoxicity, they're likely doing this in a fungal specific manner. And so this also suggests that complex 2 might have some fungal specific aspects of it that could be exploited in antifungal development, which is promising since mitochondria and the electron transport chain have been heavily implicated in fungal virulence. So I've told you that we need novel fungal specific targets and how we're targeting spore germination as a fairly good target to identify low toxicity inhibitors. We can now move forward with these inhibitors and optimize them to make them more potent against fungal fungal cells and less toxic against mammalian cells, and hopefully put them in murine models of infections once we've optimized them and develop them into low toxicity antifungals. Additionally, this opens the door for potential work for prophylaxis to prevent spore-mediated disease in vulnerable patients. So the idea here is, what if we could not only inhibit the actively replicating cells, but also inhibit stress-resistant infectious papules like spores? If we could inhibit both, we might have a chance to prevent spore-mediated disease in individuals who we know are most at risk of invasive fungal infections. And with that, I want to thank you for your time, and I want to thank the whole lab for their endless support as well as funding mechanisms. Thank you.
Great. So Sebastian, great talk. We have a couple of questions about the bimodal class example. So the first one comes from Matt Blango, who says that the bimodal class example reminds him a bit of bacterial persister cells, where you have the small group that's metabolically inactive while the others grow. And so he's wondering, if you collect the spore population and then move them to fresh media, will they again show a bimodal phenotype, or will no one terminate? If you look at that, say either extreme.
That's a wonderful question. I think the power of this assay is that these phenotypes can tell us how different inhibitors might be working. And when we first saw a heterogeneous germination phenotype, I was caught off guard. My thought was, okay, so all these spores, there's something that makes them different. They're not a unique cell, like they're not all the same. They're not plano. So the bimodal phenotype is interesting. And I've always thought of it as part of the population is killed and the other part of the population is not affected at all. And so one of the things I'm currently working on, or that is being worked on in the lab, is coupling the assay with live dead staining to see whether that population is dead. Purifying down the spore population after the germination assay would be tough. I'm sure we could figure out how to do it, but my current hypothesis is that they're dead. That may not be the case. I hope that answers your question. I think it's tough to say right now, but it's definitely an intriguing phenotype that it's not on the spores are clearly not equal, but it's a bimodal response. So what is so different about spores A and spores B that makes them either fully germinate or not germinate at all? And the first thing that comes to mind is they're dead, but I'm not sure. Yeah.
So I also had a question about the bimodal phenotype, and that was, does this potentially suggest breakthrough growth for a subset of the population? And so could you maybe even expand it to look at emergence of resistance populations, potentially weeding out small molecules that are prone to inducing breakthrough populations, or just doing a general screen for how resistance evolves?
I think that's a wonderful way to use this assay. I think that there's a lot of potential there. One thing is that spores are intrinsically more resistant to most stressors than yeast. And so if you were to compare Amphotericin B, for instance, spores are more resistant. I think the question of resistance is an interesting one, because you'd probably want to make mutants that are resistant and then make the spores resistant. And the question is, will that resistance persist in a non-actively growing cell? And you can't really evolve spores. It's not like they're actively replicating. So it's not like you could do an evolutionary experiment. I guess you could evolve them, have the yeast make more spores, hope that they actually make spores, and then keep doing that over and over again. I think there's a lot of ways.
Sounds like fun.
Yeah. I mean, now that we have the tools, we might as well try it. So.
And then a question from Amelia Barber is, have you tested these inhibitors on other fungi, specifically germination of other fungi?
I think that's a logical next step. Not these inhibitors particularly. For the ones we did with the FDA-approved drugs, we did. So the nice thing about Aspergillus, for instance, is that you can actually look at the two distinct morphological transitions, right? They become larger and stay circular, and then they create a germ tube. And so one of the inhibitors, disulfiram, does this, right? It inhibits it and it doesn't create the germ tube. I haven't tested these. I think that's the logical next step, because you could imagine that you could modify each of the structural groups to be potent against a specific fungus if they were targeting fungal specific mechanisms, right? So I think that's a logical next step for these compounds.
Mhm. And then another question is, is your assay sense enough to detect something like budding in a yeast that buds asymmetrically?
Yeah, that's a good question. Not in its form that I used here. So we, in collaboration with the Keller lab, they're doing some Candida auris stuff. And so you can actually look at different sizes and shapes of budding cells. So sometimes Candida is more elongate or more circular. And so you can actually modify the assay to look at that. It's not the assay I presented here. You have to change a few things, a few parameters, but it's just size and shape, right? So you can look at this. You could look at Titan cell formation if you wanted, right? You could do all this. It's just looking at size and shape at the end of the day. So I think it has the potential for it. The way it exists for the spore germination, you can't identify post-germination budding specifically, but if you look at the cells, you can see whether they're budding or not. So
Great. Any other questions? If not, a huge thank you to both our speakers for fantastic talks. Oh, Amelia has a follow-up question, and that is, she remembers that the current SDHI fungicides show some degree of toxicity in human cells. Is she remembering this correctly? And have you examined the host cell toxicity in any of the SDHI inhibitors you characterized?
Yeah, that's a great question. So the SDHI fungicides, there are so many different ones of them. And turns out the ag industry doesn't publish most of their work with it, which is great. You know, I really appreciate that in looking for information. But our group B inhibitors aren't very cytotoxic, at least in our studies. The fungicides, it is known that some SDHI fungicides are extremely toxic, which is why they're discontinued. I think that there's a variety of reasons why they get discontinued. Have I examined host cell cytotoxicity of any of the HDHI inhibitors? Not canonical HDHI inhibitors, only our group B compounds, if they are SDHI inhibitors, which we do think they are. So hopefully that answers that.
Yeah, and the toxicity was pretty good if I remember the graph correctly.
Yeah, at least again, the way we're doing the assays, it seems like these are fairly low cytotoxicity, and at the concentrations we're testing, so, you know, some of these inhibitors are working at 2.5 micromolar and they don't show any cytotoxicity at 40 micromolar. And the thing is, the one compound in this group that is cytotoxic or shows some cytotoxicity is like the third worst. And so it's not directly linked. Yeah. Yeah, I only presented it very briefly. Sorry.
Yeah. All right, any more questions? If not, thank you both. Those were fantastic talks, and thanks everyone for coming. We'll see you again the second Thursday in August.